Hello, my name is Edgar Martinez. I am presenting protein structure and function in my educational presentation. In today's session of protein structure and function, we're going to be answering some questions as to what are proteins made of, how do proteins get their final shape, what are some examples of proteins, and ultimately we want to understand that protein structure and function leads to a greater understanding of how cells function in your body. Proteins are comprised of a set of up to 20 amino acids. Each amino acid has its own internal chemical property. The initial structure of proteins are made up of amino acids held together as polypeptides. Each polypeptide has a ordered structure in which the amino group is called the N-terminus, and the N group carrying the carboxyl terminus is called the C-terminus. Proteins can be enzymes involved in transportation and muscle contraction, hormones, cell structure, and storage, among many other functions. Polypeptide chains have chains projecting outward called side chains. Side chains are not involved in the primary structure of proteins, but side chains are what gives the proteins their unique properties. Side chains can be hydrophobic, meaning water repelling or afraid of water, or hydrophilic, water loving. The hydrophobic regions give polypeptides the structure, give the protein structure. Proteins fold into the final conformation state at the lowest energy required state. Proteins can be denatured by solvents, however, can be renatured to their natural conformational state. Chaperones assist proteins to fold into their most favorable conformation state, which is their final conformation state. In a study by Boscos and Landry in 2019, some chaperones were found to be heat shock proteins which were more expressed as a result of more heat. The two most common types of protein structure is the alpha helix and the beta sheet. A helix is composed of several similar subunits attached to each other which form a spiral. A beta sheet is a rigid, pleated structure composed of polypeptide chains which lie next to each other. Some amyloids form beta sheets that are associated with neurodegenerative diseases. And also, amyloids may also form as a result of egg fertilization to prevent polyspermy fertilization in a study presented by Egg et al. 2015. And CPLAC in 2017 found that beta sheets may bind to each other depending upon the charge of the main polypeptide chain. protein structures are made up of a primary, secondary, tertiary, and quaternary. The primary structure is the amino acid sequence. Secondary is the alpha helices and beta sheets that form within the polypeptide chain. Tertiary structures comprise of alpha helices, beta sheets, and other types of loops to form a three-dimensional structure in which extensive folding occurs. Quaternary structure forms when two or more polypeptide chains interact to become a full protein. Hydrogen bonds are key to stabilizing protein structure to prevent misfolding and equilibrate thermal temperatures. In a study by Nesis and Grisnick, 2012. Closed loops are present in all prokaryotes and eukaryotes. Era C structure in S. typhimurium is similar to Era C protein structure in E. coli, meaning that these structures are well conserved and exist along a long lineage from humans to bacteria. And this is from Lowell, Gorgialis and Hyman, 1989, and Brezorovsky in 2017. Elementary functional loops form the basic functions of an enzyme. Some enzymes contain elementary loops from ancient peptide formations. Again, stating that these are very well conserved sequences. We go back a long time. Some proteins have the ability to regulate gene expression and epigenetic inheritance. Proteins interact with glycosamine glycans, which are important carbohydrates that regulate a protein's function, such as an enzyme. Proteins can also control other proteins as ATP levels change, according to Forchhammer and Ludek, 2016. Proteins are very complex and perform many functions within our bodies. This educational program is just a fraction into the world in which proteins work to provide our existence. For more information, visit nature.com or science.com for the latest peer-reviewed articles on proteins and many other topics. And here is my bibliography page for the sources I used in my presentation.